Our next speaker is Arima Genomics' very own Senior VP of Science, Dr. Anthony Schmidt. Anthony means a lot to Arima as he's the foundational scientist behind the extremely impactful Arima Hi-C platform. He was the lead developer of the Arima Hi chip and Capture Hi-C solutions, and I wish I could share what products he has up his sleeve for 2021, but you'll have to stay tuned next year. He's well-trained, well-published, and a genuinely good person to work with. Anthony, thank you for all that you do for Arima. The floor is yours.
Thank you very much. How's my sound? Can you hear me okay?
Perfect.
Cool. Yeah, it's really great to be here. Thank you all for attending today. As it's said here in the title, and we're talking about our Arima Hi-C technology from the perspective of both sample prep and analysis. The outline today, I'm going to give a brief introduction to our company for those of you who don't already know us, and I'm also going to briefly introduce Hi-C. And then I'll talk about sample prep as well as informatics and leave some time there at the end for Q&A. It's a lot to cover in just 20 minutes, so it's going to be sort of an overview, and hopefully that'll stoke the fires for some questions that we can discuss afterwards or offline. So first, a little bit about the company. At Arima, we fundamentally believe that the genome structure is just as important or useful as the genome sequence, and we really aim to accelerate the understanding of both the genome sequence and the structure and the role in human health and disease and species evolution. And the way that we aim to do that is by facilitating access to this type of data via kits and services. Our first product was the Arima Hi-C kit. We launched that back in 2018, which you can sort of see here. Of course, as many of you know, Hi-C was really first invented back in 2009 to measure 3D DNA folding, particularly in the context of gene regulation and how the genome is organized and how that relates to genome function. So of course, Hi-C kits and services are being used by those who study gene regulation. And then as Christian alluded to, we have since launched 2 other targeted products that really capture targeted 3D genome folding information in the context of gene regulation. But as we know, and as Karen alluded to, Hi-C is really also a staple in the genome assembly workflow. So we've had many customers use Hi-C for genome assembly. The Arima Hi-C technology specifically was selected by the Vertebrate Genome Project Consortium, and we've been partnering with them for the past couple of years. Just this past year, we designed a new product. This is called the high coverage Arima Hi-C kit that I'm going to talk about today and talk about those data, really targeted at genome assembly with some improved features for the genome assembly community. And that's a separate kit that's also focused on genome assembly. And we just released that was being used by the Sanger Genome Institute for the Darwin Tree of Life project. So just in terms of the nuts and bolts for Hi-C, I mean, as a way of brief introduction to introduce it relative to conventional whole genome sequencing for, let's say, short reads, you have your cells, which you're looking at here. Really, the DNA starts off organized into a 3-dimensional space, but when you extract that DNA, typically you just sort of purify it. If you're going to sequence with Illumina reads, maybe you'll shear that, add your sequencing adapters, and perhaps amplify, and then sequence, and you sequence these short 500 base pair molecules. And I mean, that's really been a workhorse for capturing the genetic sequence for several applications, such as genome sequencing and exome sequencing. But this first step here loses that critical 3D genome organization information and also the long-range genetic information that is really intact when the cells are in this state before the DNA is extracted. So while this preserves the DNA sequence, what Hi-C aims to do is preserve both the sequence of the genome and the structure of the genome. So there's this upfront step here where you take your fixed cells, the same cells that you would start with before DNA isolation here. You go through a few simple steps. We digest the genome with a cocktail of restriction enzymes. Those ends are then labeled and then ligated back together and then purified. So essentially, these DNA that are held together in 3-dimensional space but not joined end up being joined back together. And right at that junction point, you have a biotinylated marker that allows you to enrich for that. So if you shift down to sort of the library prep side, it looks very common. It involves adding sequencing adapters and whatnot. But the key difference is that, like I said here, you're able to enrich for those ligated pieces of DNA. And then what gets carried through the whole library prep process is these shown here, gray and blue molecules that are held together in 3D space. And you can actually now sequence those on the same read pair. And when you can align those back to the genome, that's essentially a single data point that measures that the gray piece of DNA and the blue piece of DNA were spatially proximal. And the conventional NGS data has very short inserts, like I said, about 500 base pairs. And what's unique about Hi-C is that these insert sizes, or the distance between where these map, is really dictated by the folding of the genome. And you can have sequences that are hundreds of megabases away, 10 megabases away, hundreds of kbs away, this entire distribution. And that's essentially like a ultra long linked read, if you will, that's useful for scaffolding and phasing and some of the other concepts that Karen introduced. So like I mentioned, we were part of the VGP project, but this slide is really just to set the stage for how the Hi-C technology sort of fits into the assembly workflow. We talked about PacBio reads as well as nanopore reads and other approaches for scaffolding. And here's sort of where the Arima Hi-C platform fits into phase 1 of the vertebrate genome project. And of course, there's lots of active development of other assembly workflows that are actively ongoing. So sample prep, very specific to Arima Hi-C. What I wanted to do in this section is actually sort of transition from the Hi-C, Arima Hi-C introduction that I had in the beginning, and put this sort of in the context of the high coverage Hi-C data that I introduced sort of in the beginning, and the high coverage kit. So first, some motivation. I think that there's always this sort of feedback loop between bioinformatics analysis and how that can improve sample prep, generate data, analyze it, and then this sort of constant feedback loop. And that's what we did really for the development of the high coverage Arima Hi-C kit. So using some of the vertebrate genomes, this was like maybe one of the first 20 that we that we generated. What we're plotting here is essentially the fraction of the genome that is accessible, or the fraction of the sequence content, if you will, of a genome that is accessible via Hi-C. And that's the percentage that's being plotted here on the y-axis. And for each of these 20 different vertebrate genomes, if you use a single restriction enzyme, like in homebrew Hi-C protocols, you have access to about 70% on average of the genome, and about 30% of it you will never sequence because you're using just a single restriction enzyme, and it doesn't cut uniformly across the genome, and therefore you're blind to that sequence. In the Arima Hi-C, version 1, if you will, we used 2 restriction enzymes. That was our first product. We can get about 90% of the genome accessible from that. And this is all based on an in silico prediction. And we realized if we could optimize for 4 different enzymes, that we could effectively capture 100% of the genome in the sequence, which could be very valuable for using the Illumina style Hi-C data for applications like variant calling, base polishing, and what have you. Right, so this was based on in silico. And then if you actually analyze real sequencing data, this is a typical coverage histogram. If you look at single enzyme Hi-C data, you see this very broad distribution here. If you can see my cursor, with lots of sequence gets over 100x, and then there's also an abundance of reads or bases that are covered with no reads at all. And what you want is a tighter distribution to inform more uniform sequence coverage, and that's what you see with the purple line, which is the high coverage 4 enzyme Hi-C data. So it works in practice just like we would expect from in silico prediction. So that is the chemistry that's in the high coverage Arima Hi-C kit. This top is just the same workflow that we looked at earlier, and I just basically highlighted the difference. So it's really built on top of that proven Arima Hi-C chemistry, except that we've now optimized this particular step to use 4 restriction enzymes instead of 2 to get the improved coverage uniformity. We have some other analyses that I don't have time to show here that shows that that's pretty comparable to whole genome sequencing. It's not quite as uniform as whole genome sequencing, but it has a similar performance for some of the analyses that require uniform coverage, like variant calling. I mentioned the selection here by the Darwin project, and really retains these key benefits of the Arima Hi-C chemistry. You really want a high proportion of long-range intrachromosomal links to inform things like scaffolding and phasing. You want to minimize the number of interchromosomal links because effectively that's noise in the scaffolding and phasing process. And of course, some of the other things like PCR duplicates, robustness to sample types, et cetera. So, I mean, we're really talking about sample prep here. So I wanted to go over some of the details and about our recommendations and capabilities for sample prep. Some of the validated sample sources would, of course, be human and mouse, other vertebrates that are being run through the Darwin project or the VGP, insects, plants, and others. We work frequently with cultured cells, primary cells, FACS-sorted cells or nuclei, whole blood, fresh or frozen animal tissue or plant tissue. And we have some general sample input requirements that are listed here. I'm not going to read them off the slide, but we also have low input protocols like using 50,000 mammalian cells or less, very low amounts of whole blood, and all those protocols are available if needed. The way that we really derive these types of validated sample types is, through in-house practice, through customer validation. But I also wanted to just step through one experiment that we've done in collaboration with the sample prep group at Rockefeller, where they have sent us some zebrafinch tissue here, where our goal was that we were trying to really define a set of guidelines for how to optimally collect samples in sort of like a field setting or mimic a field setting, how to preserve those between sample collection and when you can, get them into your freezer, and what works the best in the context of Hi-C. So we have a sample collection here, and what they did for us is they flash froze the tissue immediately with no preservative, or they put it into 95% ethanol and immediately tried to freeze it, or they put it into DNA guard, which is a DNA stabilization reagent, and then froze it immediately. We also put the samples into the same preservatives, but held that at 6 hours at 4 degrees. Maybe you're out in the field collecting a sample, you have refrigeration, but you can't get back to your lab for a day. That's what that's meant to simulate. And probably the most interesting here is you're in the field, you collect your tissue samples, you put them in the preservatives, and they sit for a week at room temperature before they can go back into the freezer. And for all of these different time points, we generated Hi-C data here. And what this plot shows is effectively on the axis here, the purple is the fraction of the reads that are long-range intrachromosomal linkages, which is effectively what you want to maximize for, as well as the proportion of the reads that are inter-chromosomal linkages, which is what you want to minimize for, and that's sort of like a signal to noise. And at the fresh frozen time point, the no preservative and the ethanol here looks pretty good, but even at a fresh frozen collection, if you put it in DNA guard, you see really a spike in these interchromosomal linkages, which is not what you want. So we would not recommend using that reagent. I'm going to jump ahead all the way to the week-long time point, and quite impressively, the ethanol preservation, even a week at room temperature, you can still generate really, really nice Arima Hi-C data with these really good metrics. Of course, when you have no preservative for a week and something sitting at room temperature, the 3D architecture dilapidates. You have a lot of uninformative signal or noise. Same with the DNA guard. So that sort of helps define the optimal sample preservation and collection. So lastly, I'm going to talk about bioinformatics for a second. I'll also say that scaffolding and phasing is going to be a topic of the bioinformatics panel. Can you hear me okay? Did I drop off? Let me see.
So you just dropped off for 3 seconds there, but you're back.
I'm back. Okay.
Your screen is back.
That was weird. Okay.
No worries.
So yeah, our philosophy, I think from the onset, we've been very supportive of open source tools. As a company, we embrace the open source model. Our support scientists help with everything from pipeline installation, pipeline implementation. We share all the information needed about our restriction enzymes to inform the analysis using those open source tools. If you send us your shallow sequencing data and your FASTA file from your initial draft assembly, we'll actually subsample that data and do QC analysis and interpretation at no extra cost. So we really try to be there along the entire continuum of the project, from sample prep through analysis. We don't just support open source tools. I mean, we play a fairly active role in the collaborative development of new bioinformatics tools. This here is from Adam Philippi, Sergey Korin, Jay Geyer back in 2019, the SALSA 2 scaffolding algorithm. We have other customers who are developing scaffolding algorithms like Instagrail from Romain Kajal's lab, as well as what we'll hear about later in the bioinformatics panel, the DIP-ASM pipeline from Shilpa Garg and Hong Li et al., which is currently a preprint. Just in general, like a very basic overview of how this works. You align, say, your Arima Hi-C data to a set of contigs. That goes through iterations of scaffolding. For example, using SALSA, it outputs a scaffolded genome, and you can actually visualize that using tools like Juicebox is what we typically use. And now you can actually edit the scaffolded assembly using Juicebox assembly tools. I've seen a handful of publications, and several people find that quite useful. So, what does that actually look like? Just one single example here. When you align your Hi-C data to a set of contigs, you might see a Hi-C heat map that looks like this. Effectively, all of these off-diagonal red spots are strong, long-range interactions that are occurring between contigs, likely derived from the same chromosome. And through iterations of scaffolding, you stitch those contigs together, and the assembly becomes more continuous through iterations until you have a final iteration that has these fairly well-defined chromosomes along the diagonal here. And this is kind of what you're looking for at the end. We've had several folks, I'm not going to read off this. This is all on our website, who have attested to the Arima Hi-C data quality, the utility. A couple of these here over on the right are from the high coverage Arima Hi-C kits, speaking to the signal to noise, the low input capabilities, the more evenly distributed reads that I alluded to in the beginning, and really importantly, the robustness across a range of tissue types. You want something that works on the first try. There's a handful of publications. Karen already spoke to it in the T2T paper. The bat assembly, there are the Kita assembly, there's insect assemblies, and you can all look at these on our website if you want more information about the applicability of the technology. So in conclusion, like I said, we are super focused on facilitating access to this really important sequence and structure information. The Arima Hi-C platform is an established tool and a data type with proven performance really across a whole host of different sample types and sample preservation methods that I spoke to. Importantly, that high coverage Hi-C technology is really built on top of that Arima Hi-C chemistry, but with the advantage of the improved genome coverage. We support open source tools, and the tech support team that we have that I manage is going to be there with you from sample prep through informatics support, and we would really like to discuss your project. And with that, hopefully I didn't go over, but I'm happy to take any questions. Thanks.
Right on time, Anthony. Nice work. Nice work. We have a few questions in the chat, which is fantastic. I can see, yep, a couple more coming in. I'll start at the top and work our way down. The first is, how big a fraction of the reads end up as Hi-C reads rather than regular paired end reads?
Yeah, that's a good question. Let me step back to, oops. Yeah, I'll go to this slide. Oh, am I not sharing anymore?
Not anymore. Yep.
Sorry about that.
No worries.
So here, in these distributions here, let's take the one here on the far left. The gray bar, which is a little bit over 50% in this case, or even close to 60%, are, I guess, what you would call Hi-C reads. These are long-range linkages. The blue bar technically is also the result of a Hi-C read because those are inter-chromosomal linkages, and those could really only be formed through the Hi-C process. And then this fraction here, which is about 20%, are all of the cis reads that are less than 15 kb. Of those, if you say, what portion of them are typical, 500 base pair insert whole genome sequencing reads, it's typically about 10% less. So I would say maybe about 10% of them are just a traditional, shotgun style read. But in the whole process of, defining the coverage of the genome or, using those data for variant calling or other purposes, they're all Illumina reads, of course, and we leverage all of them for that purpose. Yeah.
Perfect. Thanks.
Yeah.
We have one more kind of follow-up question to that from the same attendee here. Have Arima Hi-C been used in environmental samples to link plasmids with the right bacterial or archaea chromosomes in soil or similar?
Not that I'm aware of. No, I don't think that we have.
Okay. Please, if the question isn't addressed, please feel free to use the Q&A box and add an additional question here at the bottom. A few more, and we've got a few more minutes, which is good. Let's see. So the next one is, Anthony, you mentioned that with 4 enzymes, the Hi-C protocol works best to achieve comprehensive genome coverage. What is the actual percent coverage you get as compared to your in silico predictions? And are there any particularly difficult sites where an extra restriction enzyme could be introduced and be beneficial?
Yeah, the way that I would say we've tried to read out like the functional performance of the coverage is through variant calling. Let me pull up in that slide here. So, I pretty much just showed a coverage histogram. I showed the in silico predictions, but one way to really prove that the coverage is in fact uniform and useful is we use the data to call variants in 4 different, in this case, human genomes. And what we're plotting here on the y-axis is the percent of variants called that were also found in the Genome in a Bottle reference set. The purple bars is the high coverage Arima Hi-C. The gray bars is just shotgun whole genome sequencing. It's fairly depth matched. You can see that compared to whole genome sequencing, we're about, in some cases, half a percentage point up to maybe 1 percentage point less sensitive, or maybe up to 3 percentage points less sensitive than shotgun Illumina sequencing. Part of this is going to be derived from the fact that Arima Hi-C data, I would say just generally Hi-C data is mapped as a single end because the insert sizes are not uniform and not predictable. So things that like variants that would be in regions where you need paired end mapping would be hard to detect currently with the Arima Hi-C v2. So that's an area where you would be blind, but that's sort of more of that's sort of more due to Hi-C specifically, not necessarily a restriction enzyme thing. We've looked at coverage in certain repeats. We've looked at how frequently the restriction enzymes cut in the centromere, thanks to the CHM13 chromosome X paper. And we do see fairly like the same number of cut sites per bin across the centromere, meaning that the cut sites are in fact present in the centromere, and it's not like we would be blind to at least the chromosome X centromere that we've looked at so far.
Okay, perfect. Thanks. And maybe just a follow-up question to that. I'll skip down a couple. Would you recommend the high coverage 4 restriction enzymes kit / protocol to be the way to go for all genomes and never mind the other lower coverage kits?
Yeah, I mean, I think so. Currently, we're in early access mode for the high coverage, high C kits, doing additional customer validation. But of course, the selection by Singer, certainly gives us a vote of confidence that it's a good way forward for genome assembly. And I think as a company, we'd be happy to discuss it on a project specific basis if you reach out to us.
Perfect. There's a question here about variation between species. Can you touch on that?
I can't see the question.
Oh, just do we see much variation between species?
Oh, I guess I've looked at this coverage. Oops, I think I'm going the wrong way. I've looked at the coverage uniformity or the accessibility in plants, insects. That's not on the slide. Vertebrates, and the restriction enzymes are sort of optimized to cut in AT-rich regions as well as GC-rich regions. So the idea was to have it robust to a different sort of a variety of different sequence compositions in the genome. But we haven't like exhaustively tested, dozens of plants or insects yet. Most of the work so far, hands-on, has been done in mammalian systems or vertebrates.
Yep, fair enough. We've got just maybe 2 more minutes. Got a question here about variant calling. How well does the high coverage Arima Hi-C data work for variant calling?
Yeah, that was on the slide that I just showed a second ago here. We looked at the sensitivity or effectively the recall compared to whole genome sequencing. And we also looked at the concordance, and it's about 99.8 or so % concordant with the gold truth calls compared to about 99% for shotgun Illumina sequencing. So that's sort of our variant calling performance metrics.
Okay. Maybe the last 2, if we have time. What options are available for higher throughput sample processing?
That's a good question. We've done a little bit of work. Maybe I have it in here. Oh, I do. Yeah. We've done a little bit of work on proof of concept automation. We've been working with Agilent Technologies and their Bravo liquid handler to try to automate the Hi-C and the library prep part. So what I've drawn here is effectively a line under the workflow where we have validated automation, specifically using the Bravo platform. We have a couple groups that are doing this in their labs now for some of the higher throughput projects, or at least trying to get this up and running. So in some, really, this part is automated, but the pre-Hi-C part, preparing tissue and whatnot, is still fairly manual right now.
We have just a couple of minutes left before we transition to the next session. I see a couple of questions here around inter-chromosomal interactions, so we'll get to those questions just as follow-up as part of this session, and wrap the Q&A up here. Thanks, Anthony, for your time. We really appreciate it, and thanks for the content.